is in the person of Dr. Derrick Edem Akpalu, and I'll read shortly on his profile. So he's a co-founder and CEO of Rivener Biosciences. His academic training and professional experience span aspects of the biomedical sciences and clinical research areas, including the role of Associate Director as Clinical Research Scientist at Johnson & Johnson's Research and Development Limited. Before this, he was a Principal Clinical Research Scientist in J&J's Ethicon Group, following his earlier role as a Clinical Pharmacology Scientist at the Janssen Pharmaceutical Companies of J&J. Prior to this, he completed a dual degree, PhD in Biomedical Sciences and Master of Science in Clinical Research at the Morehouse School of Medicine, Atlanta, USA. His doctoral research focused on post-myocardial infarction microparticle release and made the case for the use of the microparticles in understanding pathologic cardiac remodeling. He also has a master's degree in biomedicine from the University College London and an undergraduate degree in biochemistry with microbiology from Queen Mary University of London, United Kingdom. In his role as associate director, he served as steady, responsible scientist, providing scientific and clinical input to a cross-functional global team for the development, execution, and advancement of clinical strategies in the solid oncology therapeutic area. In his most recent assignment at J&J, he served as steady, responsible scientist for the confirmatory study for amivantamab, the first fully human bispecific antibody approved for non-small cell lung cancer patients with EGFR exon 20 insertion mutations. As principal clinical research scientist, he provided insight and strategic direction for innovative clinical evidence generation and dissemination strategies for new products and lifecycle management projects of the global biosurgery franchise, that is biologics and devices. As a scientist in clinical pharmacology, Dr. Akpalu's work encompassed the pharmacokinetic and pharmacodynamic aspects of biologics drug development in the immunology therapeutic area. While at MSM, he also served as a clinical research coordinator at the on-campus Clinical Research Center. In this role, he was involved in the execution of various clinical research projects in the cardiometabolic disease area, including the National Institute of Health-sponsored multi-center systolic blood pressure intervention trial. Before his time at MSM, he worked at Richmond Pharmacology Limited in London, UK, working in early clinical phase trials, including bridging Japanese studies, and was named a clinical research production manager of record on the company's MHRA license during his tenure. Derrick currently serves as a reviewer for the Molecular and Cellular Biochemistry Journal. He also serves as a fellow for Ghana Center for Democratic Development, CDD Ghana, a think tank in Ghana, and uses this opportunity to advocate for public health and clinical research advancements in Ghana. His immediate professional aspiration is to leverage the composite of his experiences and network for the attainment of Rivener Biosciences' goals of championing precision medicine and value-based healthcare solutions in sub-Saharan Africa. His enduring commitment to healthcare advancement in Africa and the world at large is embodied by his ongoing work with Rivener Biosciences, which he has strategically grown and forged many alliances for with companies in the USA, Europe, South Korea, Ghana, and other African countries. In growing Rivener Biosciences, Derrick has assembled a team of like-minded and passionate individuals who are extremely dedicated to bringing much-needed healthcare innovation to Sub-Saharan Africa using access to accurate diagnosis as a starting point. It is my honor to introduce Dr. Derrick Edem Akpalu, who is coming to present on the topic From Lab to Life, Strengthening Biomedical Research in Cancer Studies. Doc, please, you can share your screen and begin your presentation. Thank you.
And so for me, and that's what I tell my patients, ivermectin.
Hello, kindly grant me, okay, hold on. Share my screen. Okay.
Recommend again. I don't.
All right. Hello, everyone. For being here. About research quite often in Africa, we publish and we attend conferences. Yet the issue that we want to talk about, or we at Revna Biosciences focus on mainly, is the and so what of it. What are we doing? How are we intending to solve these issues? Because as we sit here right now, there is a patient sitting in the waiting room, Confanoche or Kolebu, or UCC, or any other location that one may think of, waiting too long for a diagnosis. And they're still receiving, in some instances, treatments that are designed for someone else's biology. Today, I want to talk about what it actually takes to close this gap, what we think it takes to close this gap operationally. I am Derrick, Edem, Akpalu, as mentioned, Revna Biosciences. And I'm going to share with you just directly what it is that we're doing as an entity to this. So my talk's going to cut across 6 main areas or sections. We're going to start with the problem we're looking at solving, and then we'll move to who we are and why we think accreditation is central to that. And then we go deep into 3 pillars of good cancer research. And then we'll look at how all of that connects into a model that actually moves science from the lab to the patient, because ultimately that is what we care about. How do we get all of this to benefit the patient? And then I'll leave a little bit of time at the end, hopefully for us to engage in conversations, to hear your questions and any feedback that you may have. So let's start with why this is a conversation that is urgent. Okay, let's start with the numbers. 70% of cancer deaths happen in low and middle income countries. And this is not always because of biology, okay, but it is also primarily because of systems. We heard Dr. Sabunsu mentioning some systemic issues and all. These are huge institutional challenges for us because oftentimes we find ourselves in systems that are a bit weaker in comparison to what we see elsewhere. We also have a funding gap. This has also come up in the previous discussion, as well as some of the questions here, where evidence shows that less than 5% of global cancer research funding actually reaches Africa. What that means for us then is that the science that's being done is to understand cancer that is largely being done on other patients. And we're expected to, in some instances, apply those findings here as though, biology has no reference point or zip code. And then finally, again, the lack of diagnostic penetration or the late stage diagnostics or diagnosis that we have, where and that has the most devastating consequence for under-resourced research as well. Oftentimes, by the time the patient receives a cancer diagnosis and the disease has progressed to a point where treatment options are significantly narrowed to the point where significantly narrowed. And that's a systemic failure. It isn't a biological inevitability. It is a systemic failure. So for us, this is not just from that perspective a research problem. It is a systems problem. It is a systems problem, then it can be fixed or changed. So let me tell you how Revna Biosciences is doing that. So let's start with the first thing. When you meet somebody or want to know about somebody, who are they? What's your name? Is typically where we start from. We're Revna Biosciences. The name comes from the Greek word Revna, meaning research. And that is what grounds us as an entity. It gives us that identity. This isn't some incidental thing. It is an intentional thing for us to center us, knowing that we're supposed to be stewards of good research. We're supposed to be that entity that contributes to the knowledge base of the country. In plain language, what our mission is, we exist to make precision medicine real for the African patient. And this is through better diagnostics, better data, and better science. I want to just quickly touch on our core values: research, ethics, vitality, non-conformity, and all-inclusivity. Each of this is deliberately chosen. For instance, on the non-conformity example, we refuse to accept on the right, you see just below the DNA double next to the who can know simply through learning. It is woven into a helix, and therefore it's basically, this is our philosophy made visual, saying that we are committed to lifelong learning, embedded in So talking about infrastructure and quality, what are we doing? So from a quality perspective, we think that credibility matters. We're in a highly regulated space. We're in an area where decisions impact lives. And therefore, we have to operate at a global standard, at the best quality standard that the human is capable of. So to us, delivering precision medicine at clinical grade quality requires infrastructure. It requires expertise and accreditation that takes years to actually build. It is a reason why we have invested over 3 years going after these accreditations, and not simply for the accreditation, but it is the process of the accreditation and transferring that into a living quality system that you espouse, that you live on a daily basis. Bio has made that investment, and our A2LA triple ISO accreditation is that independent verification that we have. But let's be specific about this. The triple accreditation means nothing to the patient if the service or the ecosystem within which we're operating doesn't have or doesn't feel that impact. So let's look at these ISO accreditations one after the other, and I'll weave in together why we went after these three and how we're using these for what really matters, which is patient outcome. So the first one is the ISO 15189 2022. This is the gold standard when it comes to medical laboratories. It governs how we test, how we report, how we handle discrepancies, and how we ensure that a result coming out of the lab is one a clinician can actually act on. Then we have the ISO 20387 2018, and this is the international standard specifically for biobanking. It covers sample collection, annotation, storage conditions, traceability, and long-term preservation. We had Dr. Osebus who mentioned that they needed a -20 storage freezer to store tissue samples so that they could be used possibly down the line. That is biobanking in a nutshell. And this matter is enormously central for clinical research, because biobank is where, or is the way to ensure that we have samples that are viable for future use. Then the third one being the ISO 17043. And this one is a bit interesting in the sense that most laboratories get assessed by external bodies. REVNA gets done. All these other labs participating in what's called external quality assurance. We all do that, where somebody or a qualified lab sends you characterized samples or EQA samples, and you basically have to run those assessments or run an assessment on the sample and report your findings. And the point is this: if all of us are operating at the same quality standards or at a comparable quality standard, then whatever sample sent to me, so long as it's the same sample sent to you, handled the same way, then our results should be within reasonable margin of variation, if any, statistically, and of course, with the biological context in place. So what this ISO accreditation is that now REVNA Bio is accredited to run those assessments. We can design and manage these proficiency testing schemes for other labs. And what it means is that when we are investing in this, or we've invested in this, because again, we're not operating in a silo. It isn't only, we need more revenues. We need all of our labs to be at a certain quality standard. And this is one way where we feel like we're investing in the community in which we're part of to elevate all of us. Now, let me take us into the work itself. So now you've understood a little bit in terms of the validation of quality standards that we've assigned ourselves to. Now, I want us to talk about three pillars that undergird everything that we do. Here are these three pillars. So we're talking about laboratory capacity, we're talking about research design, and data integrity. Every piece of impactful cancer research rests on these three. Because consider this: a lab that can produce reliable science. Okay, consider this. We need a lab that can produce reliable science. We need studies that are designed with rigor. And of course, the data should be a set that can be trusted. If we have any weakness in any one of these, it undermines the other two. For instance, you cannot publish credible findings from a poorly designed study. It won't happen. Even if you have a lab that is of world-class standing, you cannot publish credible findings from a poorly designed study. Same way, you cannot build precision medicine on data you cannot trust, even if your study design is perfectly structured. Now let's start with the foundation itself. What does the lab need to look like to advance cancer care from our perspective? So laboratory capacity, and we're talking about building from the foundation up. Okay, so you can't, of course, go into diagnostics and all without the infrastructure. So there are four areas here that we want to touch on quickly. We've got this facility. We equipped the lab with all sorts: digital pathology systems, biobanking infrastructure, molecular diagnostic tools, you name it. And we've then gone through the process of validating these two international standards. These are to set the tone for operational excellence or operational performance comparable to what we have elsewhere. And therefore, the A2LA accreditation that I described basically says that we have hosted external auditors in our facilities, working through our facility and basically confirming the soundness of our workflow to the international rubric. Now, in terms of workforce, of course, you have the equipment and all, but equipment without people's furniture, as somebody says. So we've actually invested quite heavily in training, building scientists and systems to help us interpret things like NGS data. We also manage the biobanking side of things through, curation, and then operate digital pathology platforms at clinical grade. All of this means that you have to constantly invest in the team. You have to build a culture of continuous learning because technology is constantly evolving, and we need to ensure that our scientists and even non-scientists are constantly on the ball learning on a daily basis, because what you know today is only as good as it was when it came out. And on a daily basis, knowledge is evolving, so you cannot be static. From a workflow perspective, as an ISO accredited entity, SOPs are things that we're used to. These are not to make things difficult for anybody, but it is more towards the reproducibility of it. If Kofi goes to the lab to do something and Amma goes in there next, both of them should have comparable results so long as they're handling the same sample type, going through the same workflow. And this is what SOPs help you do, or help us do internally. These are not only for audit and file away situations, but they are actually what govern the daily practice. Every sample, every test, every deviation from a protocol has to be documented and investigated, because we need to understand what's happening here, how do we prevent it, and importantly, how does this impact the patient? It's always about the patient. And then what is this? So from a revenue perspective, what is our approach? So we've built this modular scalable infrastructure, and because as research needs evolve, the lab must be similarly agile and be able to evolve with them. Clinical grade quality is what we aspire for, and it's not a milestone we care about because it's something that we live. Now let's move on to the next level where we look at these how the accreditations sort of impact day to day. So I want to sort of demystify the accreditation part for a moment because it's often treated as this checkbox or box ticking exercise, and really it isn't. I mentioned earlier on that for us, these accreditations serve as help us ensure that we basically have living quality systems. So the ISO 15189, for instance, is our way of ensuring that every assay we run is validated before a single patient sample touches it. We care about the sensitivity, the specificity, and reproducibility of it, as mentioned earlier. And then the standard also governs the entire testing journey from the moment that sample enters the lab to the moment the result is issued. Because again, it's all about the patient and can the clinician trust what is coming to them. On the biobanking side, for biobanking, it's all about trust over time. Trust over time. Because the question is, will a sample stored today be research grade in 5 years' time? And if it will be, how was that sample even collected? There's the ethics of it. Dr. Sebunsu mentioned institutional review boards and all of it. All of these things come into play here. And for a sample to even be viable for use in 5 years' time to answer burning research questions requires that chain of custody to be maintained. We need precise annotation of these samples and should be able to put these samples in storage that is controlled with the necessary traceability as well. And this is what the ISO 20387 for biobanking helps us. It's that framework that guarantees that these outcomes are possible. And then mention the ISO 17043. As I mentioned, we run the proficiency testing scheme. And this means that we design these external assessments that other labs use to verify their own performance. So look at it like, almost like a peer-to-peer, but this is one where one of those peers is accredited to be able to run accredited proficiency tests. And that is a responsibility that we take we take very seriously, and it demands its own level of competence, and that's why we had to go through that training. But in the midst of all of this as well, what is important, another piece that is important, are those research partnerships. No one institute or entity can do it all. And the practical implication for anyone listening right now is that no one institution can do it all. You can have the biosamples, data sets, and diagnostic results, and that's coming from an entity, but you may have a gap that needs to be filled. But importantly as well, the work that is being done, is it publishable? We have to ensure that it is clinically also actionable and that it can withstand the peer review process. So quality samples, quality infrastructure, they are necessary, but they're not sufficient by themselves. The study design applied to the infrastructure has to be equally rigorous. Next, let's talk about the research design. Now, good research always starts before the first sample is actually collected, way before first patient is sampled. A good research would typically have started, and it usually starts with a clearly articulated hypothesis. Then you justify your sample size so that you have that generability potential if that's embedded in the long-term goal of it. Then you specify your endpoints and have your documented controls so that at the end, whatever it is you're publishing is valid, it's got the publication characteristics. But then, our context, community-centered design is another area that we need to look at, because typically research conducted on African populations, or usually if you have a study that is conducted on a population without the community being genuinely partners of it, you're ethically compromised and scientifically weaker. Because representation in study design is beyond the equity argument. It is a validity argument in its sense. Then we mentioned about sample size justification, and also we sort of hinted at the statistical integrity of it. Pre-registration, power calculation, those kinds of things, transparent analysis pipelines. These are not in there as bureaucratic hurdles or inconveniences, but they are actually what make the difference between finding that replicates and one that does not. What we're trying to do is we're building a research culture that treats statistical integrity as something that is non-negotiable from the get-go. So we also ascribe to standards. And again, part of the earlier and laid the foundation for this, where we use these frameworks to ensure that we're operating at a standard that is globally accepted and comparable. But we also do recognize that the African patient data requires its own reference infrastructure, which leads me directly to the next slide, because the African cancer data is not just different in its volume, it's also different in its character, and that difference matters. So we pay attention to that. So here we're asking, why do we think, or why is the African cancer data different? There are three key points we want to talk about. The first one being the genomic diversity, that we have a more genetic diversity than any other continent. And therefore, a variant that may be clinically significant in a Ghanaian patient may not even exist in data sets or databases used to interpret genomic findings in Western labs, and vice versa. So the key point is this: if or when we apply the wrong reference, we risk misclassifying variants, and in cancer diagnosis, a misclassification could literally lead to or cost somebody their life. Then there's the cancer biology difference that we're paying attention to. With TNBC, triple negative breast cancer, presents at a higher frequency in West African women, according to literature. Cervical cancer mortality disparities in Ghana are not primarily, or they're driven by differences in cancer biology, but they seem to be also driven by access to screening and treatment. So our research designs must account for these realities, or we'll be solving the wrong problems. Then there's the consent, trust, and participation angle that needs to be also considered. We understand the historical issues, that there's a history of research extraction on this continent, right? We have studies where data and samples have left Africa, publications have followed, and communities saw no benefit at all. For us, we think that building genuine research partnerships requires acknowledging that history and then earning this trust through transparent and reciprocal engagement. That's what we mentioned earlier on. This community engagement, very important, especially for those of us who care about cancer care on the continent, because this directly affects recruitment, retention, and the validity of your findings. Now, I'm going to transition to the third pillar, which sort of holds all of this together, because none of all of this is useful if we cannot trust the data that we're generating. So data integrity, more and more, we keep hearing about this. The data is not, or is only as valuable as it is trustworthy. And in precision medicine, what we say is that data that cannot be trusted isn't useless, or isn't just useless. It is actually dangerous, because direct impact to someone's life. Structured data architecture. We've used standardized ontologies so that the data can be interoperable. Dr. Osebunsu mentioned some data interoperability issues or challenges that they've witnessed, and this is a real problem, not just in Ghana but globally. So all of us have to find ways to tackle this, because we need to make sure that our data are or is interoperable across systems and interpretable across time. It's not just across systems today, but in 10 years' time, could you come back and then interpret those? Could you even retrieve them? Audit trails become important. We have things like version controls, because the key point there is what gets stored should be retrievable and readable in 10 years' time, and not just for today. And then we have the regulatory and ethical compliance bit that was mentioned. So for us, we ascribe to the Global Data Protection Regulation for governance. IRB oversight is non-negotiable, and participant de-identification. We do all of this to ensure that we're protecting resources to producing science that is valid. Makes participants consent, and often some of the colleagues take sight of that. Next, I want to talk about the translational research gap that we're trying to solve. See, across the pipeline from left to right, we have discovery, validation, translation, and impact. In a well-functioning research system, these stages flow. They flow from left to right, 1, 2, 3 to 4. Okay, findings from the lab will inform clinical studies. Clinical trials generate new evidence. Then the evidence changes guidelines. And then these guidelines change patient outcomes. Standard of care evolves and all of those things. But here's where the gap is in most African countries. The pipeline breaks down somewhere here from discovery to impact. And once again, I'm referencing first speaker, Dr. Osebunsu, mentioning that they have instances where research findings are sitting somewhere collecting dust. That's because of that gap between discovery, between discovery and impact. Who is being that steward, making sure that the information that or those insights are actually translating into what really matters or getting to where it matters most. It isn't about publication. It's about outcomes. It's about improving standard of care. It's about the patient. Okay, so this is where we like to call it the Revner Bridge, because that is what we believe, or what our response to that gap is, being deliberate and multi-pronged, where we're building, or we built an integrated biobanking that creates sample assets for validation studies. We have accredited diagnostics to produce results that can inform clinical guidelines. AI decision support tools to get those insights to clinicians at the point of care, and policy engagement to ensure that what we learn actually influences how cancer care is delivered in Ghana. Because the goal is for patients to survive because of what we published. So let me show you how all three pillars and accreditation connect into that single coherent sort of model. So this slide basically is just to synthesize a lot of what we've said. So I'll take it a bit slowly here. So we've talked about laboratory capacity, producing clinical grade biosamples and diagnostic output that researchers and clinicians can actually trust. And that trust is the prerequisite for research design to generate those findings worth publishing, and data integrity to ensure that those findings can be interrogated, shared, and then built upon. Okay, now you see right here that spine from lab capacity to research design to data integrity. We're talking, this is running beneath everything we do, or running beneath all of this research capacity, research design, and data integrity. This is where the ISO accreditations come in. That is the quality infrastructure that makes all of this credible. If you take the accreditations out, each of these pillars exist on their own, but the guarantee disappears. And therefore, this is a coordination of these to ensure that we're solving for that cancer care problem in a way that makes sense in our context. And here's what that system may look like operationally for us. So this is what we've built, and this is what we're working on currently. So we We're building that a tumor and liquid biopsy repository that is ISO 20387 compliant, linked to clinical metadata. And this is the foundational asset for cancer research in Ghana. Samples that researchers can actually access and study. Then we have the molecular diagnostics bit of what we do. We have everything from full blood count all the way to NGS-based, anything in between. We have NGS-based oncology panels, digital pathology workflows with rapid turnaround for clinical decision making. See, Dr. Osebunsu mentioned that again. Duration. When he was training or they were training, it was taking 9 months plus in some instances for a biopsy result to come back. Now we're down to 1 to 3 weeks. Fortunately, Revna is pushing that turnaround time down by the day as we go. So hopefully in near real time, we will have some of these insights to ensure that the most important part of all of this equation, the patient, is impacted today, right now. Then we have the AI diagnostics. Pathology AI trained on African tissue data sets. See, again, we can't borrow from other populations and expect them to work for us. So we have to go through that training process. Partnerships and multidisciplinary teams looking at these training modules and asking questions from their vantage point, so that collectively we can build models that make sense in our setting. So decision support tools that are designed for oncologists working in these are resource constrained settings, and building the data sovereignty from the start. Oops, sorry. But we can't do this alone. I think I mentioned that earlier on. So here's where we need to amplify a little bit, and we're talking about if cancer care is to be done well, how some of these things that all of us need to chip in and do. Okay, the research infrastructure and that Ghana needs, it's not something that any one institution can build. It requires that, deliberate effort, trust-based collaboration across. We've distilled it to about 6 key partners or partnerships. So we have the teaching hospitals, obviously, it's where the patients go, samples originate, and where the clinical context lives. So it's a part of this. We have the international networks that we need to care about, because these help with validation cohorts. They oftentimes, we've talked about money during the first presentation, and funding comes from some of these international networks, and being part of that, the network of these international scholars helps build that peer credibility as well. Then we have the medical education bodies, where and students, for instance, those on the call today, they are today basically listening and participating, but importantly, is realizing that these are going to be the oncology researchers and clinicians of the next decade, and therefore engaging with these bodies, engaging with the next generation now is beyond the common sense. It's the succession planning thing that needs to happen. We need to invest in that, and we're doing that through co-ops, internships, project support in our own little way. But then there's the broader biotech industry as well, that we need to tap in, engage, because when it comes to things like even procurement, knowledge transfer, joint validation, it's about looking and making those partnerships and harnessing everyone's perspective, so that we all fully understand the African context and ensure that what we're doing, what all of us collectively do, leads to the most important part, which is impact for us or for our patients. Then there's the regulatory and policy bit that we need to pay attention to, and oftentimes we do engage on a daily or weekly basis or periodic basis. Ghana FDA, Ethics Board, the Vaccine Institute, you know, and just to understand that compliance is really, it's the pathway for our findings to influence national cancer policy. We need to be able to demonstrate that. We need to be able to do that and understand regulatory or regulation, not as bureaucratic inconveniences, but those are the assurance that all of us should seek, that these things have conformed, have been done to the regulatory requirements, that we've demonstrated the discipline required to be stewards of research, to be stewards of the opportunities we have to impact and influence, patients' life. Then we have the diaspora and the development funding. It's the Ghanaian diaspora expertise and capital. We can do without. We've all talked about it. There have been questions here. How do we fund this? How do we get paid time off to do XYZ in the cancer research part? We may have to look at some of these avenues because, again, government alone can't do it. Foreign donors may not be so aligned in terms of their funding priorities. Therefore, that the knowledge and investments that exist, how are we going to harness that? What is needed is a trusted vehicle to deploy them, to find and deploy those opportunities. And that is part of what we're trying to build from our side. So what does this part actually require? And we'll just be very quick on here. I think we're almost there. So the path forward is long, is slow. The siloed research model, one hospital, one PI, one grant, it's not what we need. It isn't. And if you look at how Revna is looking at it and trying to solve this, it's a reason why we as a clinical stage precision medicine company, we're doing biobanking. We're doing clinical research, not just as practitioners, but as service providers as well. But then we're also doing diagnostics and even playing in the access to medicine space as well. Because the siloed model of research is not capable of generating that scale of evidence that changes cancer care outcomes at that population level. It will not happen. We need co-designed multi-institutional research agenda that is not, that is how that science can change guidelines, or science that changes guidelines gets done. From a sustained and diversified funding perspective, international NGO funding is time limited and agenda shaped, like I mentioned on the previous slide, by donors who may not be Ghanaian. So we need institutional grants, diaspora investment, and public-private partnerships that are built around Ghana's own research priorities. Okay, we also want to focus on African-centric reference data sets. We cannot deliver precision medicine to African patients using built for European cohorts. It will not happen. We will lose out. Building African reference data sets is not a nice kind of thing to have. It is the scientific prerequisite for precision oncology on the continent. We also need that culture of translational urgency. Urgency, not tomorrow. Don't be promising the tomorrow's patient. What about today's patient? That is what we say in Revna. And that's why we don't only do research or support research. We also care about investing in diagnostics today, because we believe that in solving today's patients' diagnostics challenges, we're better positioned to help tomorrow's patients. So let me close with that thought, and let us sit a little bit at the center of everything that I've shared today. And that is simply this, okay? The quality of research determines the quality of our clinical decisions, okay? And therefore, strengthening biomedical research, it shouldn't be seen as an academic process. It is a life-saving work, and we need to see it that way. Everything I've talked about up until this point, the accreditation, the biobank, the study design framework, the data infrastructure, it all exists in the service of one outcome, that a patient in Ghana receives a diagnosis that they can trust, a treatment designed for their biology, and a care path informed by science that reflects their reality. Revna Bio is building that infrastructure for the future, but we cannot build that alone, and we shouldn't even try it. Okay, I think I will end my presentation with that. Thank you.
Thank you very much, Dr. Edem Akpalu, for this insightful presentation. You've mentioned a lot of things, but one that I actually took is publishing a paper is not the real impact, but the impact is when a patient survives because of the knowledge generated. And this is on a premise that most people have the idea, if you don't publish, you perish and all of that. I think this is very important as researchers to take notes. All right, thank you very much, Doc. So please, the floor is open for questions as Doc gets ready to answer them. So please, if you have a question, you can, and you wouldn't want to talk, you can type it into the chat box. Okay, Fortune, please ask your question.
Thank you so much, Dr. Akpalu, for that insightful talk. Yes, one question we have to keep asking is, what about today's patient? I mean, when we meet at these conferences, we are always talking about the problems, and we are talking about what needs to be done for the sake of future patients. But we have to take into consideration today's patient, because people are actually suffering from cancer diseases that have, they have cures, but the Ghanaian patient is still, struggling, is still dying from curable cancers. And one thing I want to confirm or clarify is, it's a multidisciplinary team. There is a role of the laboratory scientists or the laboratories, and also the role of the clinician. I just want to ask, what is Revna doing to make sure that the clinician is abreast with, the services or the infrastructure that is available to make sure that their patient gets the right diagnosis, gets the right treatment? That is my question. And I'm sure, aside publishing of your research, what other steps is Revna taking to make sure that the clinician who sits in their consulting room is abreast with current trends when it comes to cancer care? Thank you.
Thank you so much for that, Fortune, and a very good question. So what are we doing from a clinician engagement perspective to ensure that, we're in step or lockstep? Now, a couple of ways we're addressing this. If you take a look at a typical Revna oncology report, you'll notice that our reports are a bit more annotated than the alternative. And this is intentional because we understand that part of what we do or what we're doing is not just about pushing reports out. It's about educating the clinician as well, or making available to them in real time new evidence as it becomes available. It's also about shifting the standards and making sure that we're all evolving because we're in a very dynamic state. So you'll notice that even basic things to just draw people's attention to a few things are on there. You will notice that on our reports, we actually show sample adequacy data. If a sample is sent to us and we're not able to extract adequate nucleic acid from it, we have to make sure that that is noted. If we're able to do it, we note it there as well. And you even make a comment about it. And why are we doing that? It's not to show off, it's to draw the clinician's attention to something that is outside of their clinical practice, but that has big relevance. Sample adequacy, hovers around the information or the state of the sample at the time it was collected or during transit to the lab. It is important. And this is one way we're trying to draw people's attention to that, clinicians' attention to that. So our reports are annotated quite heavily because we see part of our role in this service as being the scribe for the physician or the oncologist, being a scribe. So we provide a bit more literature information and some context as well for their consideration based on the clinical context and the patient's presentation there. That's one way we're doing this on a day-to-day basis. We also have structured sessions. And we're working with some key medical authorities in country where fellowships and those kinds of trainings happen at the REVNA facility. And this is important because if the person who's out there farming doesn't realize all the available tools they have, they might be using a machete for something that a hoe may do, or may not even realize that they have the machete close by. The point is this: by having the clinicians rotate through our labs or spend some time with us, they get to see the tools we have. They get to understand from a technology perspective what the use case for each of these could be, and are able to then discriminate between requesting a particular test for one patient and requesting another for another patient that may be presenting similarly to the other, because they understand the subtleties of the technology and undergirding the reports that they're asking for or the request that they are submitting. We're also working on webinar series and all. Internally, we have a marketing communications person that's helping us put out some of these educational content out, and you'll be seeing a bit more from REVNA Bio in the days and weeks coming. We've also brought in a clinical and scientific liaison whose role purposely is to be that bridge between our clinical labs and medical fraternity, to be able to speak the clinician's language and translate that into the clinical operational setting that we work in. So these are some of the ways that we're trying to ensure that the technology that we have, the findings that we have, and the friction points that we even have are made known to the clinician.
All right. Thank you very much, Dr. Akpalu.
Thank you very much, Fortune, for your question, and Doc for the answer. So please, I have a question from Ernest Edward Samoa. He's asking, for the AI-driven cancer diagnostics, pathology AI is being trained on African tissue data sets. Who owns the intellectual property of the algorithms developed from this data, and how do we ensure data sovereignty remains in Africa? Thank you.
Great. So this is a question all of us are solving. We're trying to solve for in diverse ways. It's a reason why we need that joint collaborative approach, because one person wouldn't have all the answers or know it all. What we're doing is looking and making sure that from a standards perspective, as demonstrated, we are applying ourselves to the highest rigor available because it is dynamic. And what is acceptable today may not be acceptable tomorrow. So that is one approach we're using. Data ownership, usage, and IPs. These tend to be quite project specific. I have seen instances or research situations, or I've seen situations where the IP is more communal, given the funding source, the research design, and all of those. So it tends to be quite, in some instances, project specific as well. But I do acknowledge that there is a gap that needs to be filled in terms of the IP ownership and part of things. Now, in terms of data storage, data integrity, and data sovereignty aspects of it, there are basic things that at the institutional level we can do. How are we safeguarding our data? Are we encrypting the data in real time while storing? What sorts of cloud systems are we using? Do we use on-site server systems? And if it has to be in the cloud, where in the cloud is it sitting, and in what format? So digital solution steps that can be taken at the institutional level. What I will say about data sovereignty, though, is beyond the obvious, what we see, like what I've mentioned and all, is the gap where we need to advance for us. Where specifically data protection rules had to keep up with the changing landscape. They're doing what they can do, but I believe that collectively we need to support a bit more from that perspective. But then there's, from a functional perspective, one of the concerns that we also have from a revenue perspective in terms of even data sovereignty is this practice of samples going overseas for testing. That's a big gap there. And you have that conundrum to address. People need tests. Now, if the capacity isn't available locally, then what happens, right? But in instances where those capacities can be locally built, what are the incentives? Or if we notice those gaps, what is the incentive to ensure that those capacities are invested in locally? These are some of the subtle components that we need to look at. And I thank you for bringing that question up.
Okay, thank you very much. Please, the very last question, and Ernest was doubling down on his question regarding workforce development. He said you mentioned training scientists from biobank curation to NGS interpretation. That's next generation sequencing. Are you finding a sufficient pipeline of local talent, or you are relying heavily on the expert expertise to bridge the skills gap? Thank you.
Thank you, Chu. And Ernest, another very good question. So, Revenue Biosciences invests very heavily in ourselves, as in the scientists and team members that we have here. We're constantly undergoing some form of training and all. There is always a gap because we're a very ambitious entity, and we're always taking on more. So there's always that sort of tension or lag in terms of the available skill set and the projects and interests that we want to solve. How we're going about this is, for instance, using things like national service to bring in service personnel to rotate through the facility, and at the end of their rotation, should we have the slots available, we retain those that need to be retained. For instance, what we're also doing in some instances is using collaborative models to sort of outsource certain elements of it locally, or in rare instances internationally for a short period of time while we build capacity locally. But I must say, though, that because that focus has been front and center for us over the last 3-4 years, we've had team members go out on company sponsored training to even European destinations to get trained or to gain skill sets that they then bring back and become trainers of trainees for us. And that's how we're sort of bridging these gaps. The other thing is what we do is every new platform we bring, we ensure that the tech transfer happens, the technology transfer happens. So the manufacturer or the owner of that technology takes us through training, and we ensure that we are capable of operating this platform or technology in-house. And it just doesn't happen only on the application side. It happens on the biomedical side, because bringing these equipment in brings a huge risk with it, in the sense that what happens on your preventative maintenance side. Yes, you can skip on that for a while, but that only means that that equipment is going to break down pretty soon. And what happens when it breaks down? So we invest in biomedical engineering training as well, so that we can ensure that these equipment are working. But then through that, we're capacity building as well. I hope that answers your question, Ernest.
Yes, it says that. Thank you very much, Doc, for your time. We appreciate you taking time off your busy schedule to present on this wonderful topic. Thank you very much. So we'll move on to the next and final presenter, and Fortune will introduce the